Hello, friends. This is Dr. Murphy here. I'm so excited to show you a short video of my lab. I'm going to take my phone here and walk around my lab and explain a few things. So this is my research lab. I work here at Augustana with undergraduate students on research, getting them involved in projects, and helping me publish papers and write for grants to support the research. I also help students with research in my classes, in the labs. So my research is focused on bacteria and specifically genetics of bacteria.
Do they call you Miss Brooks?
Yeah.
Okay.
Alrighty. So, well, thank you so much for joining our, virtual classroom today. This is my kiddos, I guess. This is my bio professor. Her name is Dr. Murphy. I'll let her introduce herself and then I'll ask her some of your questions, but we're going to get right to business because we don't have a ton of time together.
Hi, friends. This is Dr. Murphy. I'm a professor here at Augustana. I actually had Miss Brooks in class last year. Super, super, super awesome person. So glad you have her as a teacher right now, and super excited to get to join you today.
Awesome. So we're going to just start with those few questions that we have on our list. And the first one is, how many years of college did it take you to get where you're at today?
Yeah, so that's a great question. So to be honest with you, it was a lot of years, but it was worth it. It's what I wanted to do. So right after high school, I went straight into college for my 4-year degree, which is a degree, an undergraduate degree, bachelor's in science. And then from there, my senior year, I applied to PhD programs and was accepted out in Washington State at Washington State University to complete my PhD in genetics and cell biology, and that took 6 years. So that was 10 years total, which is a lot, but again, it was really worth it.
And that's 10 years after high school and middle school and elementary school. So that's 10 extra years. So I don't want to hear any of you complaining about school.
And then actually after my PhD, I went out to New York and I was at a university for 2 more years. So that to do a specialization kind of training. So that's 12 years on top of middle school, grade school, and high school.
Yeah, so I should not hear my students complaining anymore about that.
No.
Do you enjoy your job? That's another one. Do you enjoy your job, or is your job fun?
Absolutely, yeah. I actually don't consider my job a job because I get to have fun every day. So every day that I show up, I get to work with students, so students like you all. So the students here, though, are, of course, older. They're 18 to 22-year-olds. But what I say is I get to work with college students every day in the classroom, in the lab. I travel with them to other countries. I work with them in athletics, in different clubs. So that's what I do pretty much every day from when I get here until when I go home. And even in the evenings, I'm communicating with them.
Yeah, for sure. This is kind of a specific one. How do you, and they put you in all caps, help our world and our environment?
Yeah, really good question. So for any of us to help our world and help our environment, we have to be educated. And education comes in different forms. But part of being educated is educating others. So as you have information that you feel would be useful for others to help them, help the world and the environment, then you share that with others. And that's really the best way that I can think of that I help our world and environment is I, again, educate others and share information with others.
Absolutely. This is one of my favorite questions. What's your favorite thing you've done as a microbiologist?
Yeah. Gosh, when I saw this question, there are so many cool things that I've got to do as a microbiologist. But one that really comes to my mind right away is when I was in New York. So after I finished my PhD, like I said, I went out to New York for 2 years. I actually worked at a university, but I was also doing research for a pharmaceutical company. And this pharmaceutical company was specifically trying to get metabolites or chemicals out of bacteria that they with hopes of using those as a treatment for breast cancer. So it was really interesting because I got to work with a pharmaceutical company, which is different than academia. And I also got to be involved in medical research and specifically drug therapeutic work. So super
That is
cool.
so cool, especially with groundbreaking cancer research. And not a lot of people can say that they've
Yeah.
worked on, part of the cure, which is so cool. I guess that kind of leads us to our next question. What is one thing that you want to do that you haven't done already as a microbiologist?
Yeah. So the cool thing about bacteria, my favorite thing about bacteria, and I think someone else asked that, is the fact that bacteria can live everywhere. So I think there was a question also about sloughing off of your skin and your clothes, and will it get on you, and can you feel bacteria? So we can't feel bacteria unless they harm us, and then we don't feel well. But they're everywhere, bacteria are everywhere. And it's really good for us to expose ourselves to microorganisms. We have to. We have to build our immune systems. We have to build our guts, our microbiomes. So bacteria are everywhere. And so imagine yourself going into like a deep thermal vent or living out in an Arctic like ice glacier for years. We couldn't do it, right? So, but bacteria can. And so gosh, what I would love to be able to do is to go and collect bacteria from some of those really um, different and harsh environments, extreme environments. But I'm not sure if I could survive in some of those environments. So again, I don't know if I want to go down into a deep thermal vent and see what happens.
Yeah, you might go down, but you might not come back.
Exactly.
That's the scariest part.
Yeah. My cells wouldn't like it.
So this next question, what's the most interesting bacteria you have studied?
The most interesting bacteria is actually the one that I work on, which is called Myxococcus xanthus. It's a beautiful bacterium, but it's not pathogenic, so there's no harm for me to work on it. But its genetics are very similar to a bacterium called Pseudomonas aeruginosa, which is involved in cystic fibrosis. So if we can better understand the organism that I work on, then we can better understand the organism that causes harm to people who suffer from cystic fibrosis. So.
Yeah. And so with that bacteria, have you done any cool experiments with that in the lab? I know we'll see your lab video after this interview, but have you done any like super cool things in the lab?
Yeah. So a lot of the work we do is genetics. So hopefully someday you'll get to study more, and you probably already have at least heard of genetics. So we all have a bunch of different genes, and bacteria have a bunch of different genes. And so what we do is imagine, 3 genes, and we completely get rid of one of the genes in the bacteria. And then we ask, how does this bacteria behave without that gene? So, if I took out the gene out of you that allowed for your eye color to be like the pigment that gives you eye color to be produced, if I took that out of you, then what color would your eyes be, right? Would you even get a color? And so we ask those kind of questions. So it's really cool to just be able to take an entire gene out of an organism, because it's very hard in humans, but it's pretty easy in bacteria. So.
Very cool. And I think you've kind of talked about your favorite thing about your job, but if a microbiologist wasn't your job, what other job would you have?
Gosh, what other job would I have? Well, it was my other job would still involve teaching others, because I love the idea of educating myself. I love the idea of educating others. But I also love coffee. So maybe owning a coffee shop in the mountains, in which I could educate others. But I also love to travel. I've been to every continent except for Antarctica. And so if I could somehow combine coffee and traveling and educating others, then that would be my dream job.
That is so cool. And your science background too, with the coffee beans, and you could really get into that whole sort of thing as well.
And the microbes that grow on coffee beans.
Yeah. Awesome. And this is kind of I don't know if this is a sad question or not, but were you the best scientist when you were growing up? So I know we talk a lot about like competition in our classes and how we try not to compare ourselves to others. And I think you did a really good job of answering that question on our sheet.
Sure. Yeah, I'm so glad that you all talk about competition and not competing against each other, because I think what each one of you needs to remember is that really the only person you're competing against is yourself. And so being the best that you can be, right? So maybe your neighbor earned a better grade on you on the exam, but that doesn't mean that they're smarter or better than you. It just, they did better on that, but you'll do better on something else. And so I'm not really sure how to define the best, because really all of us have the best of something that we have to offer. And so it depends on what you're passionate about, right? It depends on what your skills are, what your knowledge base is. Educating yourself can come in all kinds of different forms. Some of us are more hands-on, and maybe we would do better doing something, a job or career that's more hands-on. Some of us are more, we want to hold ourselves up and do like computer work all day, right? That's great. That's awesome. I mean, we need all of this to make the world go round. So just really making you the best that you can be. So, focusing on your education, focusing in on learning from others, and just being a good person and the best that you can be. So.
I love that. And then the last question I wanted for like in the personal question category is, what would be the best piece of advice you could give to younger scientists?
Honestly, do not give up. So it's tough, right? I know some days you struggle with classes, with schools, with topics, right? It's tough. Maybe you don't understand something. You see that it's a long road ahead of you, or maybe you didn't do the best on your exam, but that's okay, right? So just don't give up and know that tomorrow will be a better day. And it's tough, but if you don't give up, if you're passionate and you keep at it, anything in life, right? That you'll get there, right? I mean, again, look at what we talked about earlier, 12 years, 12 years to get to where I'm at, but I wouldn't give it up at all. But it took a lot of dedication and not giving up.
And so what we do is we use an organism called Myxococcus xanthus. That's a mouthful. This particular bacterium is not pathogenic, so it cannot harm you. It's naturally found in the soil. You could actually drink a culture of it and you would not get sick. I would not ever let you do that. And I do not let my research students do that, and I do not do that. However, again, it's not a pathogenic organism, so we can work on it. We won't get sick. But we do use protective measures when we're in the labs. We wear gloves, we wear goggles. We're always very careful to not spill chemicals on ourselves. So let's see. With that, I think I'll start the tour of my lab, and I hope you enjoy. So here we are in the lab. So you can see we've got lots of bottles of chemicals, jars of chemicals. We even use things such as foil that you would actually use in your kitchen. We've got things, lots of equipment such as centrifuges. We're gonna start walking around the lab here, and you can see a lot of boxes. We do have to store a lot of Petri dishes and tubes that we'll use later. We have equipment such as water baths that we can set at a certain temperature and incubate cells. We've got a meter right here that allows for us to test the pH of a solution. And we have sinks so that we can wash our dishes. And once our dishes are washed, they have to be put in a big oven that takes them up to a certain temperature and pressure to sterilize them. It's called an autoclave. We've got whiteboards that we write on to keep track of what we're doing and our results. As we move around, these are some of the workstations for our students. You can see they're kind of messy, but as we're doing real science, it gets messy. We have a lot of things out. We've got these little boxes here that allow for us to run DNA out on a gel that's made of agarose. So we can see certain bands, certain DNA bands. We've got ethanol around the lab for sterilizing things. We've got carboys of water that has been autoclaved, so it's clean. As we wander around here, I'm gonna go over to this side and show you our microscope. This is right here, a huge microscope that is about a $30,000 microscope that lets us take even 3D images of our cells and project them here on this computer screen. You can see up above, again, we have a lot of glassware, we have a lot of solutions, always labeling our solutions as to what they are and who made them and the date. And then we have a bunch of pipettes here. So these allow for us to measure very small volumes of solutions, like 1,000 microliters, which is less than a milliliter. And then I know this equipment here looks old, but this equipment right here allows for us to take measurements of our cells. So what we do is we grow our cells in solution in a flask. This happens to be a Clet flask. And then we can dump the solution into this side arm and place it in here and get a readout of the density of our cells. And then once our cells are at a density that we need, then that's when we take them from the liquid culture onto a Petri dish. This particular piece of equipment here allows for us to use really high powered sonic waves through our cells to really bust our cells open and let out the different parts in the cell. This right here is an incubator, and it allows for us inside to incubate our plates on the shelf, and then down here we can incubate flasks and we can shake them. We also have various kits around the lab that allow for us to get DNA out of our cells. And we have this, I think is really cool. This happens to be a microscope that we use that was printed on a 3D printer. Pretty cool, with a little computer attached to it. And then we have centrifuges. So these we can put our little tubes in, put the lid on, spin it down really fast. Let's see here. We have a whole bunch of tubes, racks of sterile tubes. We always throw our trash in containers like this and then autoclave them to kill the bacteria. And so I mentioned earlier that we basically take our DNA from those liquid cultures and we come over here to the refrigerator, and actually right now we don't have any plates I can show you because we don't have students working in the lab right now because of COVID. But naturally we would put those on a Petri dish and spot them and look at the cells using that high-powered microscope that I showed you earlier. So again, I hope this gives you an appreciation for what the lab looks like, and hopefully someday I'll see you as a student at Augustana and maybe you can work in my lab with me.
Saw the video from your lab. What are some tools that you use in the lab? I know we'll look at those, but if you want to talk about maybe your favorite ones.
Yeah, so I'm glad you got to see my lab. I wish that there would have been students in there who could have also talked to you, but right now with COVID, we're not really operating. And we didn't have Petri dishes out that I could show you beautiful pictures, but you get to see, you'll get to see a few pictures on a poster from my lab. But we use pipettes, which are tools to help us measure small volumes of liquids. We use a lot of different really cool glassware, beakers and flasks and little tubes. We use centrifuges, lots of different media, food stuff for our bacteria, pretty tasty stuff. And then just, I think, like as far as equipment goes, I showed you microscope. So that's one of our favorite tools, right? Because we can't see what we're working on just with the naked eye. So we have powerful microscopes, we have little microscopes. I think, and then the piece of equipment is called a sonicator that basically kind of blows the cells open so that we can open them up and get out what we need to from the cell.
Yeah, and my students know we did a little microscope lab, but these microscopes that Dr. Murphy is talking about are very, very, way more high tech than ours are. So those are super cool. What is, do you know off the top of your head the highest magnification that you can get?
Probably 100. I'm not exactly sure, to be honest with you. We use a 40x, but see already the microscope, like the big one that we use, that's a $30,000 microscope. Already, like that 40x is different than the 40x on the more compound microscope. So that it's a hard comparison. Yeah.
Just the fact that it's a $30,000 microscope tells you a lot. I would not let my sixth graders even look at that with your sticky hands. But that's awesome. Would you say is the best part about working with cells?
They're super easy to grow. Yeah, so I can grow a liquid culture of a bunch of bacterial cells in a couple of hours. Whereas, if I were working on mice or plants or some organism that had to grow, for many months, of course, that requires more time. So it's easy. We can work fast.
Yeah, that is really good. And my students have talked about that too. We talked about the difference between mice in experiments and bacteria, why we would use mice instead of monkeys and cells instead of mice. So we can, we've talked about all of that. So that's really exciting. Do you have any really funny stories from lab, or have you ever had a really bad accident in the lab? We talked about lab safety a lot in our classroom.
Yeah, lab safety is so important, my friend. So I'm glad you talked about that. Anytime a student wants to join my lab, that's the very first thing we cover is how we deal with safety in the lab. And so for us, we wear goggles, we wear gloves when we do our experiments. We always clean up with our benches really well when we're done. We have things like a fire safety, a fire extinguisher and safety shower. I've never had to use those, and I never want to because we're just really careful. But let's see. So has anything bad? Really, nothing bad, and nothing's blown up, nothing spilled that, caused a burn. I did have in graduate school, there was another graduate student who, for some reason, didn't want to believe that you should not microwave foil. So this particular student would put foil on top of their beaker of water and put it in the microwave, and then the microwave would start smoking. And we all would remind the student that you're not supposed to microwave foil. And then they would do it again about a week later. I don't know why, but don't microwave foil. And as far as funny stories, so that's kind of a funny story, but the funny stories really come in just the stories that my students tell me. And I think your teacher, Ms. Brooks, can talk about this too, just some of the funny stories we would tell in class. And I always ask students to share stories and jokes. And so those are some of the best stories. Yeah.
That is awesome. And then the last kind of question about analyzing samples. So we took We did 2 different labs. We did a potato as the Petri dish lab. So we swabbed different places in the classroom. We also swabbed our cheek cells and looked at those under the microscope. And so we did kind of those 2 methods of analyzing samples. What is your favorite way that you like to analyze your samples in the lab?
So, actually, honestly, the experiment that you all did where you looked at your cheek cells, that is honestly one of my favorite experiments. I love doing that experiment because I love seeing my own cells, but I love also doing that experiment with students because, again, you get to see your own cells and you can clearly see them under the microscope. So, I mean, of course, I love working with bacteria because that's what I do in my research lab, but seeing those cheek cells is so cool. So I'm really glad you all got to do that.
Yeah. And then I think it was really cool too. So my students know that I'm not lying. One of the questions was, what is the worst bacteria to have in your body? And you answered that. And that's the same one that we talked about. So that C. diff bacteria is something that is very bad. That's what we looked at in a case study with the patient. So they learned about a case study. And I think it's awesome that you confirmed that. Yeah, C. diff is crazy. Have you ever seen C. diff in a microscope?
I have not. No, I haven't worked with C. diff. And as a kid, one of my dreams was to work with really kind of nasty bacteria. And now I could, but I wouldn't want to work with students with it. So
that's a bad good. And then I guess other ones, we talked a lot about good bacteria. Do you know any really good bacteria to have in your body?
So lactobacilli. So you've heard of lactose and milk sugar and lactobacilli. So keep your eyes on when you go buy yogurt and you look on the side and it says active culture, look and see if it lists certain bacteria. And I can guarantee you that those are super good bacteria for your gut microbiome.
Yeah, that's awesome. And the gut microbiome is what we talked a lot about with our fecal transplants.
Yes.
And I know Augustina doesn't really do a lot of fecal transplants, but have you ever been at an institution that did?
So no. So larger universities that have large research labs are going to be the ones that do work and also are associated with a medical school. So for example, University of Iowa, they do fecal transplant work. So some of the research labs there are going to collect fecal samples and then work on fecal transplants for different diseases. So, but at small colleges, it's really hard because it's human matter, right? Yeah. So to get the proper kind of clearance to work with it, it's really hard unless you're a large research institution or a pharmaceutical company.
Yeah, that makes a lot of sense.
Yeah.
And then I guess the last kind of question I have is we are ending our microbiome unit and we're moving into metabolism and body system. And one of the things is about cells and communication. And I know we talked a lot about that in microbiome in college about how cells communicate. Do cells communicate with each other?
Absolutely. Yes. Every day, all day. So what cells do, so imagine one cell and a second cell is they produce little chemicals that they send to another cell. And then that other cell recepts or picks up those chemicals and then does something, right? So imagine you send your friend a signal to say jump, and then they pick up that signal and then they jump. Right. So, and so cells, so for example, with metabolism, maybe the cells receive a signal that there's glucose present, right? So sugar, good sugar. I love sugar. And so your cell picks up, oh, glucose. Now I need to start metabolizing it. That's food stuff that I can use to produce energy. So I need to do that. So I'm very excited you're talking about metabolism.
Yeah, and I am excited too. I don't want to give too much away because that's going to be our whole next unit. But I think that leaves us with a great place to end. Thank you so much again for taking time out of your day to talk with my students. I think they're going to be really excited to see all of the things that they've asked for their questions.
I'm so excited that you have the teacher that you have, and I'm so excited I got to join you. And maybe, hopefully, someday I'll get to see you. And maybe I'll get to see you at Augustana as a student.
Yeah, for sure. Well, thank you so much.